Hey guys, Sarah here. So today I'm going to go over the third video in our corn snake genetics series, and we're going to go over dominant corn snake mutations. If you are not 100% comfortable with Punnett squares, how to make them, how to read them, go ahead and go watch the very first video in this playlist. I will link it in a card above and probably in the description down below as well, so that you can go and get a feel for Punnett squares before you start this video. Also, you'll probably want to watch the second video in the series that I made last week on recessive mutations, because recessive mutations are going to be the most common ones that you'll run into with corn snakes, so you're definitely going to want to be comfortable with recessive mutations before moving on to dominant mutations. The dominant mutations that we're going to go over today are toffee, buff, and tessera. For those who may be unfamiliar, toffee, buff, and tessera are the only known corn snake mutations that are known to be dominant. Now, there is another mutation known as mask that a lot of people have probably heard of that's on the dominant corn snake mutation list on Ian's Vivarium. I'll link that in the description down below. Super helpful website if you guys are trying to review some of this stuff that you don't want to like go through all the videos every single time. It goes over the list of all the different morphs that are out there and different combinations, stuff like that. But on Ian's Vivarium, mask is in the dominant mutations. But through my own breeding trials and the breeding trials of other people that I know, mask has actually been considered more of an incomplete dominant mutation instead of a dominant mutation. We'll go over incomplete dominant mutations in the future, but for now I wanted to just go over the ones that we know for sure are dominant, and that's going to be toffee, buff, and tessera. Now I'm going to use T to signify that the locus has the gene that we're talking about, and I'm going to be referring to T as tessera through all of this because tessera is probably going to be the most common one that we're going to be working with. Buff and toffee are a little less common, and it's also not 100% clear if buff and toffee are completely different genes or if they're the same gene, or other things that are going on. I can talk about that later in the video, but for now just remember that T means that the mutation is present on that allele, on that locus, on that side of that snake's DNA. And X, if you'll remember from my previous videos, means it's just a placeholder. It means that there's nothing there. So I'm going to start out here with pair 1 and kind of give you guys a little bit of a refresher on how to fill out these Punnett squares. Pair 1 is going to be a tessera that is homozygous tessera to a snake that is not a tessera at all. So I'll go ahead and fill this out for us. Remember, these T's for the tessera go down, and then the X's go across. So I'm going to put the T here, and then this T here, and then X's across. So what you see here is you have 100% het tessera. So if you remember from the previous videos, het or heterozygous means that the gene happens on one side of the DNA but not the other side. And here we have 100% of them have it on one side and not the other. And if you recall, in the previous video we had anery, and when we bred anery to a normal that did not have anery, we got the same result. We got 100% of the babies that had anery on one side and nothing on the other. And so here we have 100% of snakes that have tessera on one side but not the other. So 100% het tessera. Now, a het tessera is different than a het anery in that it is dominant, and that's what a dominant gene means. When the tessera is on one side but not the other, it still shows. And that's why it's called a dominant mutation. A dominant mutation is going to be a visual mutation even when it's only on one side. So het tesseras are still going to be visual tesseras. There is no known visual difference between a heterozygous tessera and a homozygous tessera. So it's called dominant because when the gene is on one side and not both, you can still see it. And you can still see it in its full entirety. It doesn't look like it's mixed with normal, or it doesn't look like it comes in patches. It's 100% visual, the gene. And we will go over incomplete dominant and co-dominant in a future video where this still kind of applies, but it's not a perfect system. But as far as dominant mutations go, dominant mutations can be fully, completely visually seen, even if it's only on one side. It does not have to be on both for it to be seen. And that is why it's different from recessive. So I'm going to go ahead and explain the second pair that we have over here. We have a het tessera, which by the way is going to be visual, to a non-tessera, a snake that does not have any tessera at all. Now remember, if you can't see tessera, that means it isn't there at all. It's not going to be like amel, where if you can't see it, you can breed it to, another amel and you might or might not get some. No, if you can't see a tessera, it is not there. If you can't see the gene, it is not going to be there at all. So we're going to breed a het tessera to a non-visual tessera. And as you can see by going through the Punnett square, we have half that are not tessera at all on the bottom here, and then half that are het tessera. So remember, these het tesseras are going to be visuals, and half of them are not going to have tessera at all. Now, a lot of people will look at this and say, oh, well, one of the parents was a tessera, so that means that these normals might carry tessera. No, tessera does not work that way. It is, if it's there, it is there and it's visual. If it's not there, it's not going to be there at all. So just remember, it's different from recessive mutations where, oh, if one parent shows it, that means some of the babies carry it. It's different from that because if it carries it, it's actually showing it all the time. So if your snake has a tessera, has the tessera gene at all, you're going to see it. If you don't see it, it's not going to have it at all. We're going to go on to pair 3 here, and pair 3 is where it gets kind of interesting. So here we have a het tessera to a het tessera, which again are going to be visual tesseras with tessera on one side and not tessera on the other side. And what we get here is 2/4 or half het tesseras, 1/4 homozygous tessera, and 1/4 no tessera at all. Now, this is what you would do if you're trying to test your tesseras to find out if they are homozygous tessera or heterozygous tessera. So if you have 2 tesseras and you breed them together and you get any non-tesseras at all, that means neither parent is homozygous tessera. Same when you go through these two. This is actually, these two here are the most common things that happen when people try to find out if their tessera is homozygous or heterozygous. Now there's only a few, maybe a dozen or so known homozygous tesseras in the world, at least right now. When I say known, that means that they are proven breeders, like proven to only produce tessera offspring. And that's how you know if you have a homozygous tessera or a heterozygous tessera, by breeding them to a normal that does not have any other, like any tessera gene. It doesn't have to be a normal necessarily, it just has to not be a visual tessera. So if you're trying to produce or trying to figure out if your tessera is a homozygous tessera or a heterozygous tessera, you want to breed it to a non-tessera visual in order to find out. And if you get, 20, 30 babies and they're all tesseras, that probably means you have a homozygous tessera. But if you even get one baby out of 30 or 50 or 100 from a tessera that's not a non-tessera, then that means that your snake is not a homozygous tessera. And it's the same thing with this. If you know you have a heterozygous tessera and you breed it to another tessera and you're not really sure if the other one is heterozygous or homozygous, if you get any normals or any non-tessera pattern at all, that means that one of those parents is definitely not a homozygous tessera, or both of them actually would not be a homozygous tessera. So here though is where it gets interesting, like I said. So here we have 3 out of 4 that are tesseras, and 2 we have 3 out of 4 that are tessera visuals, and of those 3, 2 of them are heterozygous tessera, and 1 of them is homozygous tessera. So I'm just going to go ahead and put that down here. 2 out of 3, my pen is not wanting to work, 2 out of 3 are het tessera, and then 1 out of 3 is homozygous tessera. Now you might recognize this 2 out of 3, 1 out of 3 from the recessive video, and that's when we were breeding, say, 2 het aneries together to produce normals, some of which may be het anery and some of which may not be het anery. In this case, we know which snakes at least have the tessera gene. It's sort of flipped the other way around. In this case, we know that of all of the tesseras in here, 1 out of 3 is homozygous. So when you are labeling the babies, you can label them as, oops, my pen is not working today, 33% possibly homozygous, P-home, tessera. So if you have 2 heterozygous tesseras, remember that at least 33% of the tessera offspring are possibly homozygous for tessera. And the homozygous tesseras are going to be a little bit more sought out for than heterozygous tesseras. So when you're labeling any of the babies, they could potentially be sold for just a little bit more money because there's that possibility of them being homozygous. Now it's similar over here when we breed a homozygous tessera to a heterozygous tessera. So let's say you've already proven out this parent to be homozygous by doing this, and you breed it to another tessera to find out. Well, let's say you get half homozygous tesseras and half heterozygous tesseras. You get 100% tesseras, but you're not really sure which ones are which. Excuse me. The air is very dry in here. If you know that at least, if you know that you have a homozygous tessera and a heterozygous tessera, you know that at least half of them are also going to be homozygous and half of them are also going to be heterozygous. So that way you can label any of these babies as 50% possibly homozygous tessera. And so in either of these cases, you will probably be able to get a little bit more money out of these babies if you're selling, because the homozygous tesseras are going to be worth it to a breeder who's going to want to eventually get a homozygous tessera and breed it so that they know for sure they're going to get 100% tessera babies. So these are both, like I said, these are both very interesting ones because they still go through the 50% possibly X, or 33% possibly whatever. It's just, like I said, it's just flipped around. Whereas normally we would have normals that are 66% possibly het anery. Here we have tesseras that are 66% possibly het tessera. And then we have tesseras that are 33% possibly homozygous tessera. And here we have tesseras that are 50% possibly homozygous tessera and 50% possibly heterozygous tessera. But they are all visual tesseras. I'm just going to go ahead and point this one out too. If you have 2 homozygous tesseras, you will get 100% homozygous tessera offspring. But remember, if you haven't tested both parents, like back here, to find out that they're both 100%, you could get 100% tesseras, and one of the parents could be het tessera, and you wouldn't know it. So you definitely want to make sure before you label all of your offspring as 100% definitely homozygous tessera that you have done this test on both parents first to make sure. Because if you have a het tessera and a homozygous tessera, you're still going to get 100% tesseras. And if you're not careful, you could accidentally label them as all homozygous tesseras when they aren't. So just make sure that you've done all of the proper test breedings before you've definitely labeled something as a homozygous tessera. And it's the same way with toffee and buff as well. Toffee and buff look very similar to each other. They also look very similar to another possible gene that's out there called yellow jacket. A lot of people believe that yellow jacket exists, but nobody has really done the trials on it to make sure that it's sort of a real thing, or even to make sure that it's different from toffee and buff. And toffee and buff also look very similar to each other, and because of that, it's very difficult to determine if they are the same gene or if they're different. There are breeding trials underway for it, and knowing whether or not they're the same or different, that might change very soon in the next coming few years. Toffee and buff and yellow jacket, all of the above, they all reduce the reds down to kind of an orangey color. In fact, buff plus amelanistic is actually called orange because it's very, very orange colored. I prefer to just say amel buff when I'm talking about it because there's a lot of orange colored corn snakes, so I try to just not confuse people who are buying or are not sure how the gene works or what the combinations are called. So yeah, that's just to say toffee and buff. You could replace these T's, any of these T's, with either toffee or buff gene, and all of the percentages are going to be the same. It's all going to apply to those, and it would apply to yellow jacket as well if we knew for sure if yellow jacket was actually a gene. So I hope that this helped you guys. If you have any questions, put them down below in the comments and I'll try to answer them in the next video. Let me know if anything was vague or if you want me to go over something in a little bit more detail. The next video, I think I'm going to try to go over the recessive allelic genes such as Amyl and Ultra and Castagna and Sunrise, like the ones that I mentioned in the last couple of videos. So thank you for watching. Let me know in the comments what you'd like me to do next as far as morph deep dives and stuff like that. And I will see you in the next video.